Hello viewers, welcome to my channel Biology Notes. Today I will discuss about the first phase of endochondral ossification. Endochondral ossification is the process by which growing cartilage systematically replaced by the bone to form the growing skeleton. Most of the bones are formed in this process. For example, tubular and flat bones, vertebrae, the skull base, ethmoids, the ends of the clavicles, etc. So in this video, I will discuss the early stage of endochondral ossification, that is the formation of primary ossification center in the bone development. Suppose these are the mesenchymal cells which are committed to become cartilage cells or chondrocytes. These cells aggregate at the site of the future bone and condense into compact nodules. Under the influence of different fibroblastic growth factors and bone morphogenic proteins, the mesenchymal cells initially express type 2 collagen and differentiate into chondroblasts. Chondroblasts in turn differentiate into chondrocytes and proliferate to form the cartilage model of bone. The cartilage model, except where joints will form, is surrounded by a dense layer of fibrous connective tissue known as perichondrium. The cartilage model grows by interstitial and appositional growth. The increase in the length of the cartilage model is due to the interstitial growth. The increase in its width is due to the addition of cartilage matrix produced by new chondrocytes. At this stage, perichondrial cells in the mid-region of the cartilage model no longer give rise to chondrocytes. Instead, bone-forming cells or osteoblasts are produced. Thus, the connective tissue surrounding this portion of the cartilage is now called periosteum. An osteogenic layer can be identified within the periosteum. The cells of this osteogenic layer are differentiating into osteoblasts. Please note, osteogenic layer is not shown in this diagram. Because of these changes, a layer of bone is formed around the cartilage model. This bone can be classified as either periosteal bone or intramembranous bone. In case of a long bone, a distinguishing cuff of periosteal bone known as the bony collar is established around the cartilage model in the diaphyseal portion of the developing bone. The collar prevents diffusion of oxygen and nutrients into the underlying cartilage. As a result, the local chondrocytes in the mid-region of the cartilage model become hypertrophic. As the chondrocytes enlarge, their surrounding cartilage matrix is resorbed. As a result, thin, irregular cartilage plates are formed between the hypertrophic cells. The hypertrophic cells begin to synthesize alkaline phosphatase and osteocalcin, which initiate the calcification of the surrounding cartilage matrix. The calcified cartilage matrix inhibits diffusion of nutrients, causing the death of the chondrocytes in the cartilage model. With the death of the chondrocytes, much of the matrix breaks down, so neighboring lacunae become confluent or merge. Ultimately, an increasing large cavity is produced. While these events are occurring, one or more blood vessels grow through the thin diaphyseal bony collar to vascularize the cavity. Now, mesenchymal stem cells which are residing in the developing periosteum migrate into the cavity along the growing blood vessels. The mesenchymal stem cells then differentiate into osteoprogenitor cells in the bone marrow cavity. Hematopoietic stem cells also gain access to the cavity via the new vasculature and give rise to the marrow, including all the blood cell lineages. As the calcified cartilage breaks down and is partially removed, some remains as irregular spicules. When the osteoprogenitor cells come in apposition to the remaining calcified cartilage spicules, they become osteoblasts. These osteoblasts begin to lay down bone matrix or osteoid on the spicule framework. Thus, the bone formed in this manner may be described as endochondral bone. This first site where bone begins to form in the diaphysis of a long bone is called the primary ossification. All these events represent the early stage of endochondral bone formation that occurs in the fetus. It begins at about the 12th week of gestation. So this is all about the first phase of endochondral ossification. If you have any query, then please comment. Share the video if you like. Subscribe the channel for more videos. Stay with us. Keep watching. Thank you.